Hello, this is Dr. Miles Ingwers with the GreenFlow Corp, and today I'm excited to share some additional results by request about the Squid Juice product and the science behind it. So, a lot of our customers have asked about our nitrogen. So, our nitrogen is naturally occurring amino acids. So, all of our nitrogen is predominantly in amino acids, which are the natural building blocks to protein. And the reason we're so excited about this and the reason this matters to our customers and our farmers is that amino acids are basically already what biology wants. They're what the soil microbes want. They're what plants can uptake. They can also uptake synthetics, but amino acids add an extra punch. First off, synthetic fertilizers, especially things like ammonia and nitrate, these are very mobile in the water. They move by bulk flow. So you imagine you get a lot of leaching and runoff. Amino acids can be the opposite. So amino acids are wonderful chelators. And for those of you who may have forgotten what chelators are, this is basically a molecule that wraps around another charged molecule and basically takes that charge and wraps around it like a blanket and allows it to move and become mobile. So it can be taken up by a plant or other microbial or fungi. And that matters a lot when we think about phosphate and micronutrients. Basically, by having amino acids as chelators, you're increasing bioavailability of stuff already in your soil, in addition to just having an excellent source of pre-built, think about it, nitrogen, that the plant may already be looking for that amino acid, and they can take amino acids up directly. So what we have here is a breakdown just on percentile of our amino acids, basically all of our nitrogen, in a pie chart and broken down to what you can see here. And what I want to draw your attention to is that bolding there on the left. Everything that's bolded there, and some of those are some pretty big slices, like that glutamic acid, aspartic acid. I'm really excited about that. We were very excited about this because these are really good chelators. So to have 15% of our amino acid profile to be a great chelator, and then glutamic acid and 11% aspartic, we're thrilled, right? This is wonderful news for us. But it's also wonderful news for anybody who's really wants that chelation and to get the most out of our soil. So kind of jumping into why this amino acid profile is so exciting, I also just wanted to highlight what some of these amino acids can do in their own right. So there's been a lot of agricultural research on amino acids. This is kind of a hot field of feeding plants, both foliar, soil drenches of specific amino acids to really drive specific growth attributes or physiological responses. So I've got two slices here, the two biggest ones, and all amino acids do cool stuff, but these are ones that have been studied by the literature, by universities, third-party research, and just I want to give you a high-level highlight. Aspartic acid really is known for nitrogen retention, metabolism, and nutrient retention, utilization. It's also a precursor to several other amino acids, so the plants can use this to make other amino acids that they're in need of without having to de novo assemble it. So wonderful to see this in abundance here in our profile. Glutamic acid is also well known. It's basically a greening agent. So in these studies where they've done this as a foliar application where they spray just purified glutamic acid, you see chlorophyll production, greening. It's associated with assimilation. Basically, both these amino acids play big parts in these biological pathways associated with nitrogen, retention, uptake, utilization, greening. So wonderful to see this. So when we're talking about the nitrogen that our product brings, and we often have customers saying, wow, you pack a big punch for being a 222. How is that possible? Well, this is how it's possible. We're basically giving a pre-assembled amino acid that directly can go into those pathways, directly is what that plant might need, and is something that really feeds towards that nitrogen. So continuing on, just two others that I just love to talk about, just because it's cool to see some of these studies and to just give you an idea of what some of these amino acids do. So proline there on your right, this is an osmoprotectant, so it's just known to really increase when plants are under moisture stress. So for example, in some studies, proline abundance in the plants can go up literally 100-fold when this plant's under severe moisture or salt stress. So this is a an amino acid that is the plant uses basically as a part of its stress response. Methionine is a part of a SAM pathway. For those of you who are really into the science, it's a major methyl donor, and basically it converts into the plant hormone ethylene. So all of those things combined, these are really important in how plants respond to abiotic stress, stress in the environment. Basically, I kind of think of it like a vitamin C punch in the sense that this is kind of how plants respond. This is what they need. It's pre-assembled in terms of getting them ready for that response. So giving pre-stress, this is just a wonderful thing to have in the arsenal. So when we talk about nitrogen, I really do think it's important to frame things in terms of the nitrogen cycle. And for those of you who have post-traumatic stress from high school, I do apologize. And for those of you who know this like the back of your hand, I also apologize. But I like to just remind everybody, so nitrogen and nitrogen cycle. What is a nitrogen cycle? That's how nitrogen moves through the environment. And I think it's important to remember that despite the fact that nitrogen is such an important growth limiter, it's also, for crops, it's also 78% of our atmosphere. That's what we're breathing in. Yet it's in the form of N2, just two nitrogen atoms stuck together, and that's not usable for agricultural crops. That's why we put this on as a fertilizer. So in nature, nitrogen is fixed. That is brought from N2 to a usable form like ammonia, nitrates, nitrites, from the atmosphere through biological processes, or actually lightning, which is pretty cool as well, does it? But nitrogen fixation is driven by bacteria in the soil. So, and largely when we think about it in an agricultural context, legumes pop up in everyone's mind, right? The beans, the magical nitrogen fixers that have those nodes on their soil with that bacteria in it that naturally fixes nitrogen. So nitrogen is super abundant, but the usable nitrogen is what we're thinking about. It also comes from decomposition of organic matter. So that's how all of this nitrogen gets brought into the soil where it can be used. So then once it's brought into the soil, fixed from either the atmosphere or decomposition, there's this process of nitrification, which makes it more usable. You've got nitrogen and nitrifying bacteria, and then you have assimilation. That's where it gets assimilated into the plants, where it can then be re-released in the soil through decomposition, or denitrification, which this is important because I like to bring this up. And that's when nitrogen goes back into the atmosphere. And that's when basically it gets breathed out, is a way to think about it, but through these natural processes. So we did this study where we did what's called shotgun genomics. Sounds complicated, but what we really did is we just sequenced the DNA of the plants and the soil, everything around there, and got a snapshot. And we looked at all the genes in there, so that in that sample, and we said, okay, who's here, what microbes basically, how much of them, and what processes were they doing? And this is what's important because this is what we learned. First, we saw with our product, compared to an untreated, we'll call it a control, we see a 6.9% increase in plant tissue nitrogen. Now, if you're thinking, wow, from a 2-2-2 fertilizer, does that make sense? Yes, and here's why. When you add amino acids in this, let's call it healthy nitrogen to the soil, you're encouraging microbial growth. You're encouraging the abundance of microbes to increase, but also the species, the community to increase. This is regenerative agriculture. And you're kind of restoring some of these natural processes. So I'll jump into this decomposition later, but in this case, one of the big things we found when we looked at this, basically the shotgun genomics and all these genes that do these different processes, is one of the major steps here was actually being stopped. So that's why you see this squid here with that stop sign. If we go back to that denitrification, that's when nitrogen is being lost from your system. So we have all this organic nitrogen and ammonia going to nitrate, but before it gets lost back into the environment, we're actually slowing that process down so it gets retained into the soil and there's a higher degree of assimilation. We couldn't be more excited about this. We think of it also kind of like the way you would think of a bank account. First off, and I'll jump into this in the next slide, we're increasing the deposits, but more importantly, we are massively decreasing the withdrawals. So this is how we're able to drive up that nitrogen in your soil by feeding those microbes, by feeding the plants, and using this naturally occurring nitrogen in the form of amino acids. Now, how are we making deposits? That's just the last step. So this is that nitrification. There's a few microbes that we massively increase the abundance of. One of them is a Bradyrhizobium, and the other is a Burkholderia. If you're into the biological space, you'll know that a lot of growers are treating their soil with these microbes. There's a lot of technologies out there to do this. Our product doesn't contain these microbes per se. What our product does is it's the if you build it, they will come. We feed the soil such that these microbes naturally increased in abundance. That's what our data showed when we looked at this using a third-party trial in shotgun genomics. So increasing those kind of deposits into that nitrogen account, but most importantly, also decreasing those withdrawals. So that's the story we learned from nitrogen that we were just so excited to share with you. So in the former slides, I did promise I'd talk a little bit about that decomposition. So the story, though, that we're seeing here really wouldn't be complete if we didn't talk about carbon cycling in agriculture. Again, my apologies if this is bringing up miserable high school biology memories or super basic redundant, but I still do love to talk about it because it is so important. So I mentioned before that I would come back to decomposition and why that's important and in the big scheme of things and what our product does. So we saw a 9.4 percentile increase in processes associated with carbon decomposition, meaning the breakdown of matter. So in the carbon cycle, broad strokes, there's CO2 in the air, much lower than nitrogen, low concentrations, and that gets fixed into plants by the process of photosynthesis. And then as it's fixed into that plant tissue, it can then be turned into sugars. We as animals will eat it, and when we breathe out that CO2. And it goes back into the atmosphere. But the idea is that when you increase your soil carbon, you have more of that organic, basically plant debris decomposing in your soil. So that's roots from your previous crop. If you're cover cropping, there's a lot of carbon captured that way. And it's just all sitting there in your soil ready to do work. Basically, you've already made that investment in your soil nutrition bank account. What our process does is it really just helps release it through this process of decomposition. So that was a decomposition to release nitrogen that I spoke about earlier, but also we're talking both macros and micros, phosphates, all your micros are also released in this process. So this really drives soil health. And it's happening by increasing basically the rate that your soil is doing work for you. So the microbes in the soil specifically are doing work for you. Just to give you some context behind the hood, what's really happening. Microbial respiration. So that's how much it's breathing out. Imagine sitting on a couch versus taking a run, how much you're breathing out. That is increased 42.5% in your soil, or increased in our soil, at least when we did this study. And we were thrilled to see that. What's driving that, though, isn't just those microbes working harder or going for a run. It's actually the total abundance of microbes. So more food, more microbes, that's that microbial active carbon there. And what we see there is an increase in abundance, coinciding increase in soil respiration, and an increase in microbial diversity. So what species are there? What are they? Who's there? And what are they doing? We see this big increase as we kind of restore the soil microbial activity. So that's breaking down all of this stuff now that's already in the soil, all of that basically residual from the prior year's crop, roots, stems. It's breaking it all down and releasing that nutrition. We're also seeing a really big increase in evenness, which is just another sign of microbial health. So in soil that's, I don't like to use the term dead soil, but in soil that's been treated the same way, say for several years with the same crops, the same regime as far as nutrition and management, you will see eventually some of these few species start to dominate the landscape and dominate your soil microbiome. So when you have this process where you're putting all these aminos and other nutritional stuffs into your soil, you're seeing this big increase in evenness. So more species with fewer dominating the scene. A big plus here is that mycorrhizal fungi. So these are those fungi that literally grow into the roots super thin, like little hairs, and that they increase the surface area by which the roots are able to absorb water. And that is free work, and that is a wonderful thing to take advantage of. So that's a natural symbiosis, and we love to see that. That really can drive growth. If you're in the farming world, of course, I'm sure you're aware of it. A lot of groups are now, and have been, even for centuries, have been doing things to promote this relationship. We also are seeing a big increase in biocontrol fungus. So this is the idea of fungi that take out other fungi. And I'll get into diseases in the very last slide, as well as that same nitrogen fixation microbes that I mentioned before. But big picture, what's happening here? So by putting our product on the soil, what we saw in this study was a substantial increase in basically soil microbial activity. And what that did is that drove nitrogen fixation, but it really went and stopped that nitrogen from basically being lost back into the air. That was huge for us. So more deposits, less withdrawals. And also we saw that increase in that mobilization of all that nutrition that's already in your soil or in soil, our soil, from all the carbon that was already there. It just made use of what was in the soil. And that coincided with that high metabolic activity, improved nutrient cycling. And that's where you get that really resilient root system. And that's where you're able to drive that increase that we saw in foliar nutrition, so that nitrogen content jumping up. I believe that was 6.9%. And finally, the last thing we saw was a real increase in pest pressure. So in this study, when they basically do shotgun genomics, so we're looking at sequences to see what's in the soil, we didn't see root lesion nematode that we saw in our controls, or lance nematode, spot bacteria. We didn't see a lot of these things. We did see an increase in biocontrol fungus. So that's not to say, though, that these critters were gone, or that this process workly eliminates them. What it does do is it makes a meaningful impact on that soil microbial community in a way that it can police itself and be balanced. So not only does it drive that nutrition uptake, nitrogen retention, nitrogen fixation, but also it really helped eliminate some of these pests, and that's likely through natural biocontrol. I have here an image of a root parasitic nematode. So what's happening is you would see an increase, for example, in things like fungi and other good nematodes that basically control the bad nematodes. The same can be said for all sorts of bacterial and fungal type pressure. So again, we couldn't be more excited about these results. And if you stuck with me this entire time, thank you so much. And please feel free to reach out to me or the GreenFlow team if you have any questions whatsoever. Thank you.